Hello everybody. So now we're going to talk about how do you actually build a food web. Now I've talked a little bit about how we can either track energy or matter, right? So you can actually go in and figure out what individual species are eating and how many calories, like how much energy is in that food source. You can also track how much carbon there is. So, whether we're talking about tracking energy or carbon, it's basically the same thing because, we can eventually track carbon to the base of the food web where it was, fixed via photosynthesis. So that's why we can use carbon. And we can use things like trophic transfer efficiencies to find out how much of that carbon is being put into the next trophic level up. Now, how do you actually do that, though? Well, you can, look at what species are eating. You can follow a lion around and see, what species exactly that lion is eating. You can catch a northern pike in a net and see what it's been eating and see, oh, well, this northern pike here has been eating a bunch of crayfish. The thing is that really only gives you a snapshot of recent ingestion of what it's recently eaten. So what about this northern pike here, right? You can see it swimming along. We'll let the GIF reload here. All right, see it's swimming along and eats that little baby duck, right? And baby ducks are swimming all spring, right? So potentially those northern pike could be feasting on these little baby ducks. And if you go and catch a northern pike in August, well, you're not going to get any baby ducks in their bellies, right? In that gut content is not going to show that it's been eating ducks, right? Because it's only going to be stuff that was around recently. The thing is, you can get really specific data, right? You could identify these crayfish to species and figure out exactly what this is eating. And if you follow the northern pike long enough and well enough throughout the year, you would eventually be able to determine, what type of baby ducks it's eating also. But that's a lot of work. So there's this other tool called where we use stable isotopes or fatty acids. Now, this is better for assimilating over time, and it's integrative that over the lifespan, potentially the lifespan of an organism, but it's integrative over more time, but it's less precise. It doesn't give you an exact idea of what's going on. So what do I mean by this? So if you remember on Dr. Kelly Fritz's work, she was looking at fatty acids, those LC-PUFAs, right? Those were those fatty acids. She was tracking the long-chain fatty acids, polyunsaturated fatty acids, right? She was tracking them from the aquatic ecosystem into the terrestrial ecosystem. A lot of my work deals with stable isotopes, though, and so let's kind of get in with that. So what are stable isotopes, right? So if you look on the periodic table of elements, what you get is an atomic number and a mass number, right? And what this does is it tells you how many protons and neutrons are in the element. So carbon has 12. We know it has six protons and 6 neutrons. But why is it then 12.011? The answer to that is, and same with nitrogen. Nitrogen has 7 protons and 7 neutrons. Why is it 14.00674, right? And the answer to that is it's not every carbon atom has 6 neutrons. Some of them have 7. Some of them have 8 neutrons, and that makes the different carbon 12, carbon 13, and carbon 14. Now, carbon 14 is radioactive. Carbon 13 is stable. That means it's not going to radioactively decay. A carbon 13 that is created, billions of years ago is going to still be carbon 13 in billions of years ago, unless it, falls into another sun. But nitrogen 15 then is also stable, meaning it won't radioactively decay. Given though that these two isotopes then, this number is an average. So if we see 14.0067, that means very, very little of nitrogen is nitrogen 15. Very, very little of carbon is carbon 13. Even less of it is carbon 14. So what does this mean? Well, that extra neutron makes it actually heavier. Okay, so that single extra neutron makes carbon 13 and nitrogen 15 then actually react slower in chemical reactions. And in biological reactions, they also act slower. They react slower. So think of it as, a bunch of kids jumping on a trampoline here, where you have carbon 12 as the little kids and carbon 13 as the big kids. Jumping on the trampoline, and what happens? Those carbon 12s get flung off. So after the chemical reaction occurs, what you're left with is a higher proportion of carbon 13 as opposed to carbon 12. Same thing happens with nitrogen. Nitrogen, there's a higher proportion of nitrogen 15 after the chemical reaction or biological reaction compared to nitrogen 14. So this is particularly important with nitrogen. We have this fractionation that it's called between trophic levels. So primary producers are depleted in nitrogen 15, or they're light, meaning they don't have a lot of nitrogen 15. Now, the percentages are super, super small, such that they're it's hard to even show it in percentages. So we use this del 15 nitrogen per mil. I don't really want to get into that, but per mil, percent means 1 out of 100. This is the percent sign with an extra 0 attached to it, meaning it's much, much smaller there. Okay, so it's very tiny differences, but they are trackable. So you have primary producers that don't have very much nitrogen 15. And the thing is, because these primary consumers are eating the primary producers, so the herbivores eat the primary producers, and basically they have nitrogen flowing through their system, right? They're eating nitrogen in the proteins from the plants, but then they're also peeing it out in the form of uric acid or urea. So there's a net flux of nitrogen in the organism, and because nitrogen 15 is reacting slower, you basically the fish will pee out more nitrogen 14 at a higher proportion than what's actually in its body. So if you look at fish pee, it's really, really depleted in nitrogen depleted in nitrogen 15. Okay, so predators then have even a higher nitrogen 15 signature. So what you can use then is determine, you can basically just go into a food web and look at the levels of nitrogen 15 and figure out the trophic position of an organism. And you can start to build a food web there, at least a food chain here, right? So we can know that primary producers are low in nitrogen 15, then higher, and then higher. So if this predator were to be omnivorous, let's say 50% of its diet came from here, so 50%, and 50% of its diet came from here, it would appear this predator would not appear at this level within the food web. It would be a little bit lower, so maybe right there. It allows us to integrate in a very easy and without having to follow around the shark to see what it's all eating all day, for years. We can figure out what its trophic position is just based on its nitrogen 15 levels in its body. Now, carbon acts a little different. There's less fractionation between trophic levels, and such that we can almost disregard it. But with different food sources, there is a very different signature. So what we see is C3 plants and C4 plants. It's different ways in which they do photosynthesis have very different signatures. C4 plants are much more enriched in carbon-13, so we call them heavy, as opposed to C3 plants. And essentially with carbon, you are what you eat. So if we go up on the nitrogen scale, right? But we have here then this fish is going to be eating basically only this C3 plant because it appears directly above. Now, if you were to have this predator, then what is it eating, right? Well, since its carbon signature is in the middle, we know it's eating basically 50% this and 50% that. If we were to have some sort of fish here that make it an angry fish, right? Some sort of predator that is showing up here, then we would know it was only eating this, fish A and not fish B here. Okay, so what we have here then is actually a food web. We don't really think of this necessarily as a food web, but that's what it does. Sorry, I'm trying to erase those things. This depiction is now a food web, and so we have all sorts of links going on here, and we're not necessarily like depicting them as arrows here, but putting them on this carbon-nitrogen biplot, which allows us to explain and figure out what species is eating what and in what quantities. So you might be asking, like, what actually happens? Why do we see this increase in nitrogen 15? And what it has to do with is in the amino acids, when an amino acid is broken down in the body, it's transaminated. And when you take glutamic acid and basically eat it, we change it to this thing called alpha ketoglutaric acid, where we take off this nitrogen and put an oxygen on it. What we see then is any time that we're like fooling around with, any time that our metabolism and our cells are fooling around with this bond in here, we see the ability for that nitrogen signature to change. Okay, so we then, as we're building back up proteins, we will take this alpha ketoglutaric acid and turn it back into glutamic acid. And what that does then is we put on a different nitrogen, basically, and that makes for very interesting interactions with ability for that nitrogen signature to change. Whereas things like the amino acid phenylalanine won't change, because when it goes through metabolism in our body, this nitrogen carbon, this carbon to nitrogen bond here is not broken, and so what we can use that for then is figuring out, rather than we can figure out like what the trophic position is based by comparing the phenylalanine signature to the glutamic acid signature. So if you were to find a bluegill and take some muscle sample, and you could look at the glutamic acid and figure out that it's much, much higher than the phenylalanine, and it basically increases by 7.6 per mil each trophic interaction, each step. So we have the algae going to the prey species, going to the bluegill, and you wouldn't even have to like look and follow around that bluegill. You can just take a muscle sample and figure out what species it's actually eating. Right, so what we're going to come to next lecture is looking at how we can take these stable isotope and talk about them from a community-wide perspective.